Okay. Hi, everyone. I'm Brian. I'm a grad student in Todd Lowe's lab at UC Santa Cruz. And today I'm going to talk to you about tRNA genes, their local mutation rates, and a project that we just had published earlier this year that we're pretty excited about. So given that this is RNA, I think a lot of you are familiar with tRNAs at the RNA level, but I'm going to be talking about the DNA level here. So in our genomes, we actually have over 400 tRNA genes, which is maybe more than you'd expect. And just here's a map showing where they fall on each chromosome. Wherever you see a blue spot, there's at least one tRNA there. So they're all over our genomes, and we have hundreds of them. But for many of these genes, we actually have no evidence that they're being transcribed. So for example, we don't have a completely clear picture of what tRNA expression looks like in every tissue. But here's a study from 2011 that did Pol3 ChIP-seq on human livers, and they found that 224 tRNA loci were occupied by Pol3, which means that for 192 loci, they did not detect any Pol3 occupancy at all, meaning they found no evidence that these genes are being transcribed. But the liver is just one tissue, so to broaden our perspective a little bit, here's a heat map based on epigenomic data that spans over 100 different tissues. We've got tissues on the y-axis here and tRNA loci on the x-axis. And wherever you see red, that indicates active transcription, and wherever you see white or gray over here, that indicates inactivity. And to simplify this a little bit, here's the same data shown in histogram form. And across all these tissues, we find that about 25% of 107 tRNA loci are not being actively transcribed in any tissue that we have data for. And since we're working with tRNAs, this is actually very important because a lot of the time when you're sequencing tRNA transcripts, you can't tell which gene it was transcribed from. So these 9 histidine tRNA genes are exactly identical. And since multi-mapping of tRNA transcripts is especially common, this multi-mapping issue is something we really have to overcome for the study of tRNA genes. So how do we overcome it? In this talk, I'm going to show you that there actually are ways that we can tell these genes apart, even though they have identical sequences. And I'll also answer the question of, for genes with identical sequences, what's causing these differences in transcriptional activity in the first place? So we'll start off with what we know about tRNA genes and transcription and see what differences we can find between these genes. So in order to really understand the differences in transcriptional activity between these genes, we need to look at transcription as a process. So you've got the polymerase transcribing a nascent RNA here using the template strand of the DNA. But while that's going on, the non-template strand here is isolated. It's more vulnerable to mutagenesis, and we call this process transcription-associated mutagenesis. So knowing that tRNA transcripts are necessary for the production of every protein, we know that the most highly transcribed tRNA genes are going to be among the most highly transcribed genes in the whole genome. And if there's a heightened chance of mutagenesis every time a gene is transcribed, this process could play an important role. Now, looking at tRNA specifically, they have internal promoters. Pol3 starts transcribing upstream of the tRNA gene, and it stops once it reaches the poly-T signal at the 3' end, which means that the flanking regions of tRNA genes are transcribed along with the tRNA and processed out by RNases post-transcription. So they're transcribed but not part of the functional mature tRNA unit. Now, if this is a major factor, we would expect to see a lot of mutations at active tRNA loci. Here's an example of an actively transcribed tRNA locus with the gene highlighted in blue. And you can see that there are very few substitutions in the gene itself going back through these 12 species. But if we go into the flanking regions, you can see that just about every base has had at least one substitution. And a summary of these alignments is given here on top of the Philo-P score. Positive scores, shown in purple, indicating greater than average conservation, and negative scores, shown in brown, indicating greater than average divergence when looking across species. Now, if we take the average Philo-P scores by position across all human and mouse active and inactive tRNA genes in their flanking regions, color-coded according to the diagram shown, we see an extremely characteristic pattern where the active tRNA loci have high levels of divergence in the flanking regions, but their gene sequences are extremely well conserved. So to answer our question, how can we tell which genes are active and inactive when their sequences are identical? One answer comes from alignments. And we can see if a gene has been active because transcription-associated mutagenesis will have left this genomic signature. And now that we know one way of telling them apart, we can potentially come up with a model that can distinguish active and inactive tRNA loci. So here are these six of those 9 identical histidine tRNA genes from before. I'm showing you their philo-P scores here, which is, again, a measure of conservation. If I were to tell you that 4 of these are active and 2 are inactive, you'd probably guess that it's these 4. And the Pol3 ChIP-seq done on human livers backs up that claim. But since we have this set of known active and inactive tRNA genes, in order to build this model, we need to know more ways of telling them apart. So we looked at these sets of active and inactive tRNA genes and found what genomic characteristics are correlated with activity. So in addition to the sequence conservation pattern that we've already discussed, we found that active tRNA genes have much more proximal poly-T transcription termination sites. They're found in much more CPG-rich regions, and they're much closer to exons and other tRNA genes. And these are the kinds of things that we can include in a predictive model. So that's exactly what we did. So we have this pipeline called TRAP, or tRNA activity predictor, that takes in a reference genome, and also use a gene annotation file and multiple alignments if you have them. And this is up on GitHub along with instructions and everything needed to run it. And I know I've spent a lot of time talking about conservation patterns of tRNA genes, but you can actually extract a lot of relevant information from just the reference genome without looking at any alignment information. And these are the 10 features that the model uses, ranked by how strongly they contribute to accurate prediction. And 5 of the top 6 can be extracted from only a reference genome. So if that's all you have, the program can still work for you. But if available, the alignment and gene annotation information will increase the accuracy by about 5% or so. And these are readily available for most species. While most tools like this would just use information intrinsic to the tRNA gene itself, the biggest strength of TRAP is that we're also using extrinsic information, or information that comes solely from the tRNA surroundings rather than the gene itself. And this is why we can distinguish genes that have identical sequences. So because our epigenomic data is strongest for humans, we're using that as our training set, and we're using mouse as an independent test set. When we test our model, we get roughly 94% accuracy in predicting whether mouse tRNA genes are active or inactive. And to dive into these results a little bit more deeply, we're comparing our predictions to this epigenomic data for mouse, which spans 10 tissues, and our goal is to predict each tRNA gene as active if it is actively transcribed in at least one tissue, and inactive if it's not. Here we're interested in the red, which indicates active transcription. So here are the mouse tRNA loci that we predict as active, shown as a function of how many tissues they are known to be active in based on the epigenomic data from the previous slide. And you can see the vast majority of these are active in at least one tissue. And if we look at the tRNA loci that we predict as inactive, in the same way, nearly all of them are at 0, but even the ones that are active, only active in 1 or sometimes 2 tissues. When we apply this to 28 species other than mouse, here's an example of how much TRAP can reduce your tRNA gene sets for experimentation. So the cow genome has hundreds of thousands of sign elements, and most of them are tRNA derived, and tRNA-scan-SC alone has a hard time picking these apart. But TRAP can reduce the tRNA gene set down to just the actively transcribed tRNAs, and you get down to less than 400, which corresponds well with prior studies on cow tRNA gene activity. But let's get into a real example of how our findings can help with tRNA research. So let's imagine you're studying short, non-coding RNAs in primate neural tissue, and you're working with rhesus macaque as your model organism. And a quick side note, if you are interested in this, to check out poster number 514 by Alex Baggy from our lab. He's studying tRNAs in primate neurons. So if you're in this position, you're familiar with work by Ishimura et al. From the Ackerman lab and their discovery of central nervous system specific arginine tRNA that's critical for mouse neural function. And you want to discover the next one. So imagine you found this highly abundant brain-specific tRNA fragment and you've got its sequence, and now you want to do CRISPR on your rhesus macaque tissue to try to edit or manipulate the locus at the DNA level. But when you map this sequence back to the genome, you find that it perfectly matches 14 different cysteine tRNA loci. These sequences are shown here, with your newly discovered fragment shown in yellow. So there are two ways you can go from here. One is to just use brute force on all the loci and try all of your planned experiments, which would be very expensive and time consuming. Or you can, if you use trap and look at our predictions, you'll notice that only 2 of these 14 tRNA loci are expected to be active in any tissue. So you prioritize these, you save lots of time and money, and set yourself up for success. So from all this, learn that identical tRNA genes can have drastically different transcription patterns, and that the genomic environment, including GC content and the abundance of CpG dinucleotides in particular, plays an important role. We built a pipeline that can predict gene-level transcriptional activity of tRNA genes with 94% accuracy, and requires just a reference genome at the minimum. And we've applied this to 29 placental mammal species. There are predictions for over 10,000 tRNA genes, and if your model organism is a placental mammal, these might help save you time and money. So to wrap up, thanks to the Low Lab and also Russ Corbett and his lab at UC Santa Cruz. Thanks to the organizers for not only allowing me to give this talk, but keeping this conference going in spite of everything going on. And if you've got questions or comments, I think email is the best way to go. Thanks for listening to my talk, and I hope you're staying safe and healthy.